Hello everyone, my name is Lauren Marmo and I am a junior majoring in biomedical engineering. I spent my summer as a part of the Brannigan lab at Rutgers University, Camden, Center for Computational and Integrative Biology. My project focused on simulations of cell membrane deformations caused by proteins. The motivation for this project was to gain a greater understanding of the effect COVID E proteins have on membrane curvature. Coronavirus proteins are made up of multiple subunits, including the spike, nucleocapsid, membrane, and envelope E protein. It is shown that the removal of the E protein hinders the replication of coronaviruses because the resulting replicated viruses were no longer cylindrical, therefore incorrect shape, less virulent, and unable to cause further replication of the virus. However, very little is known about how the E protein induces membrane curvature. Cell membranes have a specific curvature which is essential to their function, including replication. Proteins embedded in a membrane cause specific membrane curvature. The membrane curvature is measured according to the membrane's level of hydrophobic mismatch and its deformation energy. Proteins are made up of multiple subunits which form different shapes and cause different deformations. The E protein is pentameric, made up of 5 subunits, and causes a pentagonal-shaped membrane deformation. My specific task for the project was to answer the question, if the protein shape has a larger number of sides, would it result in an increased deformation energy of the membrane? I looked to answer this question by creating a code in MATLAB that inputs the number of sides which forms the specific protein shape, then included this in the original protein membrane deformation simulation code. I was able to accomplish this using specific MATLAB functions, including in Polygon, to calculate the vertices and form shapes in a membrane. My results are shown on my poster in the form of a bar graph. I tested 5 different protein shapes on membranes with different levels of hydrophobic mismatch, positive, negative, and 0. My hypothesis was proven correct, with the exception of the circle protein which has no sides. The final results of the membrane energy values on the final simulation step show a hexagon at all levels of mismatch having an increased energy value in comparison to the shapes with less sides. My results also include the image result of my simulation with a triangular-shaped protein forming a triangle deformation in the membrane, which was true for the other shapes as well. My overall conclusion from my project was that my hypothesis was proven correct, because the protein caused greater deformations with larger shaped proteins and at different mismatch levels. However, further studies must be done on the impact of COVID E protein shape on membrane curvature. Different protein shapes should be simulated in comparison to COVID E protein to determine if a greater protein shape causes the virus to be more lethal and virulent. I would like to thank Dr. Brannigan for granting me the opportunity to work in her lab this summer, as well as Jesse Sandberg for allowing me to contribute to his research project and continuously guiding me through my work. Thank you also to Project Super, especially Kayla Fowler, for her continuing guidance throughout this program.